Cytosolic swelling. What is it and why does it matter? Cytosolic swelling is a condition in which cells expand in size due to an imbalance of ions and water inside and outside the cell membrane. It is also known as cytotoxic edema, cellular edema, oncotic cell swelling, or oncosis. Cytosolic swelling can affect various types of cells in the body, such as neurons and astrocytes in the central nervous system, CNS, or insulin-secreting cells in the pancreas. Causes of cytosolic swelling. Cytosolic swelling can result from various insults to the cells, such as trauma, stroke, hypoxia, infection, toxins, metabolic disturbances. These insults can impair the normal function of energy-dependent mechanisms that maintain the balance of ions and water across the cell membrane, such as sodium-potassium pumps or sodium-calcium exchanges. As a result, sodium and other cations accumulate inside the cell, creating an osmotic gradient that drives water influx. This leads to cell swelling and increased intracellular pressure. Mechanisms of cytosolic swelling. Cytosolic swelling involves the activation of various cation channels that allow sodium and other cations to enter the cell. These channels include voltage-gated sodium channels, acid-sensing ion channels, transient receptor potential channels, N-methyl D-aspartate receptors, sulfonylurea receptor 1. The activation of these channels can be triggered by different factors, such as depolarization of the cell membrane, acidification of the extracellular environment, inflammation, glutamate release, ATP depletion. The influx of cations also causes influx of anions, such as chloride, to maintain electrical neutrality. This further increases the osmotic pressure inside the cell. Some anions enter through volume-sensitive anion channels, which are also activated by cell swelling. The increase in intracellular cations and anions also affects the intracellular calcium concentration, which can have various effects on cellular functions and signaling pathways. For example, calcium can activate potassium channels that help regulate the cell volume by allowing potassium and water efflux. However, calcium can also activate enzymes that degrade cellular components or trigger apoptosis. Consequences of cytosolic swelling. Cytosolic swelling can have detrimental effects on cellular viability and function. Some of these effects are disruption of cellular structures and organelles, alteration of gene expression and protein synthesis, impairment of membrane transport and signaling, loss of membrane integrity and leakage of cellular contents, activation of inflammatory and immune responses, induction of necrosis or apoptosis. Cytosolic swelling can also affect the function of tissues and organs where the affected cells are located. For example, in the CNS, cytotoxic edema can cause brain swelling, increased intracranial pressure, compression of blood vessels, ischemia, neuronal death, and neurological deficits. Prevention and treatment of cytosolic swelling. The prevention and treatment of cytosolic swelling depend on the underlying cause and severity of the condition. Some general strategies are restoring blood flow and oxygen supply to the affected cells, reducing inflammation and oxidative stress, maintaining electrolyte and fluid balance, administering drugs that modulate cation channels or calcium signaling, reducing intracranial pressure or performing decompressive surgery in cases of severe brain edema. A better understanding of the molecular mechanisms involved in cytosolic swelling may lead to improved therapies for this pathological process. Swelling of ribosomal endoplasmic reticulum, is a phenomenon that occurs when the undergoes stress or damage, leading to an increase in its volume and surface area. The is a membrane-bound organelle that plays a key role in the synthesis, folding, modification, and transport of proteins and lipids. The consists of a network of tubules and flattened sacs, and the space inside them is called the lumen. The can be divided into two types, rough and smooth The rough has ribosomes attached to its surface, which are responsible for making proteins that are either secreted from the cell or inserted into the membrane. The smooth lacks ribosomes and is involved in lipid synthesis and detoxification squared. Both types of can undergo swelling under certain conditions, such as exposure to toxins, drugs, calcium imbalance, or viral infection. The swelling of the can have various effects on the cell, depending on the severity and duration of the stress. Some possible effects are alteration of protein synthesis and folding, leading to accumulation of misfolded or unfolded proteins in the lumen. Activation of a cellular response called the unfolded protein response, UPR, which aims to restore ER homeostasis by increasing the expression of chaperones, reducing protein translation, and enhancing protein degradation. Induction of apoptosis, programmed cell death, if the ER stress is too severe or prolonged and cannot be resolved by the UPR. Changes in membrane fluidity and permeability, affecting the transport of molecules across the ER membrane. Interference with other organelles that are connected to the ER, such as the Golgi apparatus, mitochondria, and nucleus. Swelling of ribosomal endoplasmic reticulum is a complex and dynamic process that reflects the ability of the cell to cope with stress and maintain its function. Understanding the mechanisms and consequences of this phenomenon may help in developing strategies to prevent or treat diseases that are associated with ER dysfunction, such as diabetes, neurodegeneration, and cancer.